DNA is a type of nucleic acid. DNA is a self-replicating material that is present in nearly all living organisms as the main constituent of chromosomes. It is the carrier of genetic information. All living things have DNA within their cells, from prokaryotes to eukaryotes and everything in between. In this video, you will learn about the structure of DNA and the replication process.
DNA has an anti-parallel structure, meaning that both strands run in opposite directions. These strands twist together to form a double helix, and the backbone is made up of linked deoxyribose and phosphates. Nucleic acids are formed from joined nucleotides, and each nucleotide is made up of a deoxyribose or sugar base, phosphate, and nitrogenous base.
Attached to each sugar is one of four nitrogenous bases: thymine, guanine, cytosine, and adenine. These bases are what connects the two phosphate strands together, kind of like a zipper. Base pairings are how the nitrogenous bases connect. Adenine connects with thymine, and guanine connects with cytosine. These are held together by hydrogen bonds. A covalent bond connects the phosphate group and the deoxyribose.
So this is a diagram of DNA replication. So DNA replication is semi-conservative since each new strand of DNA consists of one old strand and one new strand. The original strand is separated into two parts, the leading strand and the lagging strand, by a DNA helicase, splitting it into a Y shape known as the replication fork. The two strands are either in the 5 to 3 direction, which is the lagging strand, or the 3 to 5 direction, which is the leading strand. The 5 end, which is a phosphate group, and the 3 end, which has a hydroxyl group. I think that's how it's pronounced. Okay, so the two sides can be replicated differently to match with the direction they're going in. The topoisomers, I think, unwinds and rewinds the DNA strands and prevents the strands from being tangled. Once the DNA is separated, an RNA primer would bind to the starting point of each strand for replication. RNA primers are made by the enzyme DNA primase. DNA polymers is responsible for creating a new strand via elongation. There are 5 different types of polymers, ranging from 1 to 5. Polymer 3 adds 3 nucleotides in the environment onto the strand, matching them with each other, like A with T and G with C. This proceeds in the 5 to 3 direction, opposite to the leading strand. The lagging strand moves in the opposite direction, so it can only synthesize in the new strand through small fragments of DNA called Okazaki fragments. These fragments come off and reattach to the new portions of exposed DNA as the fork moves forward. The lagging strand needs a new primer for each fragment. Afterwards, the RNA primers will be replaced with DNA using DNA polymers 1. In addition, the gaps that are formed between the Okazaki fragments on the lagging strand will be bonded together through DNA ligase.
Learning about DNA is so interesting. Like we have learned, DNA contains the instructions needed for an organism to develop, survive, and reproduce. It also brings up more conversations about the future of DNA and the replication process. There are so many technological advances going on today that may help us create treatment for diseases, forensic sciences, or even finding the biological parent of a child.